Good day, class. Now we are officially done with your immunology. We're able to discuss the two arms of your immunity, which is your adaptive and your innate. And then we were also able to discuss each of its components. And then we proceeded to discussing your and covering your immune response consequences. Like, for example, if your immune system overreacts, then leads to your hypersensitivity. In or if your immune system does have immunodeficiencies, that may lead to several diseases. And now we'll move to your immunology lecture series. So it's a two lecture series for your immunology, for your serology. And the video lecture is divided into two. And I am tasked to discuss to you your immunoassays. And this is very important for us, of course, medical laboratory scientists, because as we work in the laboratory in the future, uh, we need to be equipped at least by knowledge in the lecture, because in the laboratory you will be applying this one. In or I think you done or performed this one already, the immunoassays in the laboratory. And then my uh, purpose here is to reinforce the theories behind those immunoassays. So the main purpose why we need to perform your immunoassays are for the following. Of course, we can use your immunoassays for protein analysis, like antibody evaluation. We can identify if the patient does not have or has a deficiency on a specific type of antibodies, like IgA deficiency, as we discussed in your immunology. We can also identify complement quantification. Like, for example, C3 complement quantification using your RID. I think you performed that one already in your laboratory. We can also use your immunoassays in cellular analysis and histo compatibility, such as your HLA typing and or identification of your lymphokines and or T cell evaluation. Of course, your immunoassays can also be used for the autoantibody evaluation, since the main component also of your immunoassays is your antibody or your antigens. We can identify your autoantibody there, such as your anti-nuclear antibody test. And lastly, your immunoassays are very effective and helpful, class, in identifying your infectious disease, because there are some bacteria and or viruses that are very difficult to identify and are very difficult to cultivate. So with that one, the identification via culture is not possible, then we can use your immunoassays for the identification. So you can follow my discussion using your reference book, Stevens, in the Serology Lecture Series. Uh, 1 will cover your chapter 8 and your chapter 9. So in this video lecture, I'll be covering all the precipitation reactions, and then also with your chapter 9 agglutination in your immunoassay series 2 or immunology serology lecture series 2. I will be discussing your chapter 10, which will be your labeled immunoassays, which are also very important in our profession. So again, you can follow my discussion using a reference book, Stevens. And then if you have some questions and or clarifications in this video lecture, please don't hesitate to contact your respective professors so we can clear out all your gray areas in serology. Again, I am Alfredo Hine Jr. and I'll be your professor for this video lecture. And I hope that you can get something and I can reinforce the principles that we discussed already during your laboratory session. So before digging out with your precipitation and agglutination, of course, we need to, understand, review, okay, the principles and components behind these agglutination and precipitation. And both your agglutination and precipitation class uh, uses the same components. And that is, ano class? Tama. You have your antibody and your antigen. Diba? Your antibody and antigen, we were able to characterize that one. And during your discussions in your antibody and antigen, we're able to determine the basic structure of your antibody, which is very crucial class in understanding your precipitation and your agglutination. You have your Fab here. That's the fragment of antibody binding. That's the reason why they are called Fab. And you have your FC. That's your fragment of crystallization. And then the Fab region is responsible in binding your antigen. So if the antibody will bind to your antigen, it will produce a complex, and we call that one antigen antibody complex. So in a basic class explanation, your antibody is just like a key. Okay, lock and key kasi siya class, 'di ba? So ibig sabihin ng lock and key, the key is very specific to the lock. So, ibig sabihin ng class, this antibody should be specific to a certain antigen. And then your immune complexes class are non-covalent combination of your antigen with each respective specific antibody. So take note with this one class, dapat daw specific yung antibody. But later, meron tayong mga discrepancies with the specificity of your antibody. We'll try to cover that one. And then your agglutination will produce a visible immune complex. Kaya, 'di ba, we do this one class because we cannot see your antibody. We cannot see your antigen using your naked eye. Can you see this one? No class, you cannot. That's the main reason why we will add a certain antibody to a specific antigen so we medical laboratory scientists can see the reaction. And then, so we can use a small, soluble, or a large precipitating complexes. But I would also like to emphasize on this first slide class na not all agglutination is a positive result because later I will also introduce to you a agglutination inhibition principle where the positive result is no inhibition. Ayan, para hindi lang talaga kayo malito. Okay? So, naiintindihan naman 'to, 'di ba, class? So, meron tayong antibody, may antigen. Pag dumikit, merong merong antigen-antibody complex. Then, therefore, we can see agglutination and or a visible immune complex. So, you have two words here, class, specificity and cross-reactivity. Kasi 'di ba, pag sinabi mong specific, dapat hindi na siya maghahanap ng iba kasi dapat specific na siya, 'di ba? But there are some antibodies that are also has cross-reactivity. So when we say specificity class, this is the binding site of your antibody directed to one. And take note with this one class, one lang daw na antigen are not complementary to another dissimilar antigen. So, ibig sabihin class, supposedly the Fab region is specific to the epitope. Diba? Dapat epitope ng antigen should be specific to the paratope of the antibody. Di ba naalala class during the antibody-antigen discussion? You have your epitope. The epitope is found in your antigen. You have your antibody, which is found, which is your paratope, which is found in your antibody. Then your paratope will bind to the epitope. Tama? That is specificity. However, class, there are some instances that the antibody has a cross-reactivity. Okay, so this occurs between bacteria that possess the same cell wall polysaccharides as mammalian erythrocytes. So I can give you one example class. Okay, like for example, in your Mycobacterium tuberculosis, di ba meron tayong BCG? Did you know that your BCG, alam niyo naman di ba class kasi you discussed this one already in your bacteriology. So in your Mycobacterium tuberculosis, yung vaccine natin is BCG. But what we use there, the antigen is correct class. We use your Mycobacterium bovis because your Mycobacterium bovis class is part of your Mycobacterium complex. Di ba tatlo yan sila class? You have your Mycobacterium tuberculosis, Mycobacterium bovis, and Mycobacterium africanum. Those three Mycobacterium species, they are categorized as one as your Mycobacterium tuberculosis complex. Ibig sabihin, halos magkahawig sila in antigenic structure. Kaya pwede natin gamitin si Mycobacterium bovis as a substitute for Mycobacterium tuberculosis for the vaccine, because your bovis is, although it can cause infection to human, but it can easily be cleared up. Okay, compared with your Mycobacterium tuberculosis. Yeah. So, ayun, nagkakaroon tayo ng cross-reactivity. Ibig sabihin, class, the two and or three bacteria na almost the same yung antigenic structure can produce an antibody that may also neutralize the three. Kaya nagkakaroon tayo ng cross-reactivity. Okay, so I hope that you understand, diba? And then we can connect what we learn in your bacteriology, kasi this is the beauty class of your immunology. It will cross every professional courses that we are taking during your third year and your fourth year. And this subject will try to give you a whole appreciation of the beauty of our course. I hope that since you'll be finishing all your professional courses now already, I hope and I pray that you can correlate everything now already. And then, the interactions naman, class. Okay? Kasi diba, sir, ang sabi mo kasi dito, sir, the antibody will bind to the antigen. So, the binding class here, ito lang, the simple binding, we call that one affinity. Okay? This is the initial force of attraction between a single Fab region. Diba? Fab region is the fragment of the antibody binding site of the antibody. Ito yung kamay niya, class. Kita niyo to? This is the Fab region of the antibody molecule and a single epitope. Pag sinabi kong epitope, class, that is your antigen. That is part of your antigen that can bind to your Fab region. Tapos pag tinanong naman kayo kung anong part ng Fab region, class, that is your paratope. Diba? Naalala during your antibody discussion. So pag dumikit to class, okay, with your antigen, specifically your epitope, to your antibody, specifically to your paratope, then we call this one affinity. That's the initial force of attraction. Sir, bakit nagkakaroon tayo ng initial force of attraction? Okay, according to your book, you have a lot of forces, class, okay, or interactions that contribute to the initial reaction or attraction. So we have hydrophobic bonds, hydrogen bonds, van der Waals forces, at saka electrolastic forces, class. Okay, so when you say hydrophobic bonds, these are major bonds formed between the antigen and then the antibody. So pag nagdikit daw yung antigen natin, class, at saka antibody, using your hydrogen bonds, then nagkakaroon tayo ng initial interactions. Then the side chain interacts and exclude water, kaya nagkakaroon tayo ng hydrophobic, diba? So, ine-expel niya. Okay, exclude the water molecules from the area of the interaction. Diba? Ine-expel out niya yung water. Diba? Just like your mycobacterium. Diba? Bakit sila nagka-club? Because they are hydrophobic bacteria, class. Diba? That's the reason why they clump in the in a certain, for example, colony. Okay, kasi hydrophobic sila. So the same with the interaction with this one. Okay, so ini-expel out nila yung water so that mas merong interaction. Okay, result in a gain in energy and form an energetically stable complex. Okay, that's your hydrophobic bond. Also, aside from your hydrophobic bond, you also have your hydrogen bonds. Your hydrogen bonds can result from the formation of your hydrogen bridges between the appropriate atoms when your antigen will bind to specifically the epitope, class, that will bind to the paratope of your Fab region. So, meron tayong major hydrogen bonds. Okay, so yeah, meron tayong O. Okay, bind to a single bond to your H, then O. Or oxygen, oxygen, nitrogen to nitrogen, and oxygen to nitrogen. Next is you have your van der Waals forces. Although this is a non-specific attractive forces by the interaction between your electron clouds and your hydrophobic bands. Okay, your van der Waals forces, we explained this one already during your biochemistry. And this is one of the main reason why nagkakaroon tayo ng affinity with your antigen and your antibody. So, next term that we will discuss is your affinity. Okay, kasi kanina, you have your affinity where there is an interaction or initial force between your antigen and then you have your epitope. So, next is that we need to measure the uh, strength. Okay? The strength of the binding ng epitope mo at saka affinity. Hindi pwedeng holding hands lang sila, class. Okay? So kahit na tawag nito, a lot of external environment or factors that may influence the uh, dissociation of your antigen and your antibody, kung malakas yung avidity niya, class, hindi siya natatanggal. Okay, so when we say avidity, this is the combined strength of an antibody with its antigen. Okay, so for example, this one, pag dalawa yung nakabind with the Fab, then it's more stronger. Okay? Compare with the one. Kasi pag one, possible talaga na marupok eto, class. Gamay lang na, maliit lang na problema, then they will dissolve. Okay? And dissociate. Compare with the avidity na strong yung nila, interactions. Of course, pag madami yung antibody that bind to a specific epitopes of your antigen, then the avidity is more stronger. Kaya, class, according to your book, sabi niya ni Stevens, that the low level of your affinity can be compensated with high affinity. Okay? So, if kung masyadong mababa yung affinity mo, class, then you can increase that one using a lot of antibodies that can bind your specific epitope in your antigen. So let's move now to your precipitation, class. Okay, so before we will jump off to your precipitation, allow me to introduce your zone of equivalence. Okay, so here, this is your zone of equivalence, class, where your antibody, okay, the number of your antibody is equal to the number of your antigen. So if kung meron tayong equivalence, class, of your antigen and your antibody, then you can observe your complex formation or antibody complex formation. So the zone of equivalence is the optimum, okay, a number of your antigen and your antibody forming a complex. And then we also have a zone of antibody excess, and we call that one prozone, class, okay, where there is an excess of your antibody. Then the antigen binds with only one or two antibody molecules. So, ibig sabihin, class, masyadong marami yung antibody, kukonti lang yung antigen natin. So, in this case, we cannot form a complex, then therefore, walang antigen antibody complex. So, wala tayong makikita, class. Okay? Next, we also have your post-zone, class. In your post-zone, there is an excess of antigen. So, take note with that one, class, ha? Kasi with your pro-zone, there is an excess of your antibodies. Now, in your post-zone, there is an excess of your antigen. So, masyadong madami yung antigen, class. So, every available antibody here, okay, site is bound to a single antigen. Supposedly, one Fab region will bind to a specific antigen, and then the another one should bind to another one. Kaya nagkakaroon tayo ng complex formation. However, ang nangyayari kasi dito, class, kasi masyadong madami yung antigen, the antigen will bind to the two Fab regions, not allowing to form your complex or no cross links are formed. So that's the main reason with your post-zone. Diba? So in order to resolve this one class here with the zone of antibody excess is we can dilute the antibody here if the antigen is the unknown. Okay. Also here with the zone of antigen excess or post-zone, if the antigen is the unknown, we can dilute the sample naman here. Okay? So, with here, also pala class, with the zone of antibody excess, if the antibody is the known, and then the antigen is the sample, then we cannot dilute, right? We cannot dilute the antigen kasi kukunti na nga lang sila class. Then therefore, either we can ask the patient to collect again after 1 week so that the antigen are more expressed. Then we can go to your zone of equivalence. Yun lang naman talaga yung purpose natin, class. Okay, we need to observe and meet the zone of equivalence. So, sir, what would be the implication for us medical laboratory scientists if we don't know this one, the zone of equivalence, the zone of antibody excess, then the zone of antigen excess? Okay, this consequence class is that uh, for example, if you collected if you collected the antigen, if the antigen is from coming from the patient, then you collected this one sa patient natin class too early with infection, what will happen? It can cause a false negative result. Okay, so in mind, as a medical technologies, kasi alam natin na, baka possible nasa zone of antibody excess pa siya. So what I need to do is, maybe I can talk to the physician, doc, maybe the patient is still under the zone of, or it's still under the pro zone. So it would be better to recollect again maybe after 2 days or 3 days so to confirm if kung zone of antibody excess pa talaga siya or negative result siya. Okay, so remember class, we're not just in medical technologies where uh, we only manipulate the machine. But now we are medical laboratory scientists, then it is more emphasis on the scientist part. Then we need, okay, to contribute, okay, to the patient's management. Okay, so that's our main job naman, class. So I hope that you understand this one because this is very crucial in understanding your precipitation in your agglutination. So, sir, I have one question kasi. Kasi, di ba, yung precipitation in your agglutination, the end product is still the same. There is a visible complex, correct? So, the difference between this one, because you need to memorize this one because it will appear in your quiz, in your exams, in your pre-board exam, and your board exam. So the difference between this one class is that when you have your precipitation, we are dealing with your soluble antigens. Okay? And then if we discuss about your agglutination, then we focus on your insoluble antigens such as your red blood cells, bacteria, and or antigen fix on your beads. And later we'll try to discuss this one. Okay? And then, although the result of antigen-antibody interaction is the same, whether agglutination or precipitation, and then therefore the similarity is cross-links antigen in your that will form your precipitate. Okay, so agglutination, insoluble antigens, and then precipitation, you have your soluble antigens, insoluble, soluble antigens. So allow me to go back with my early slides here. Kasi nilagay ko naman siya dito, class. This is extracted from your reference book. When you say precipitation, this is the soluble antigen with soluble antibody to produce E-insoluble complex that are visible. Diba? This is our primary goal. We need to observe the complex. Compare with your agglutination, this is a particulate antigens. Okay, for example, you have your red blood cells in our bacteria to form a larger complex. Okay, when a specific antibody is present. Okay, so I hope that you can understand the difference between the two. Anyways, either by precipitation and or agglutination, the end product is still the same. Diba? So, dapat makita natin yung reaction class. So, let's move and let's start with your precipitation class. Okay, so precipitation involves the combination of your soluble antigen with the soluble antibody to produce a visible insoluble complex. As extracted in your book. Okay, the simplest methods of detecting antigen and antibody reactions. So I'll be introducing three precipitation methods. We have your passive immunodiffusion, you have your electrophoresis, and you have your light scattering. So let's start with your passive immunodiffusion class. Your passive immunodiffusion method is in which a concentration gradient is established for a antigen or antibody. And then diffusion of the reactants to form your antigen-antibody reactions without your electric current to speed up reaction. Kaya siya tinawag na passive class. 'Di ba? Kasi we allow the movement of your antigen and your antibody in a natural way. Okay? So, hindi natin siya ini-induce via your electric current. So, you need to memorize that one kasi 'di ba passive immunodiffusion. Okay? So from the word diffusion pa lang class, you know the idea already na magdi-diffuse siya. Ano yung i-diffuse niya? Immune na part. Ano yung immune natin dito? Class, you have your antigen and your antibody. Paano siya nagdi-diffuse? You have your passive. So ibig sabihin class, hindi siya mediated with any other forces such as your electron. Okay? So of course, the movement or the diffusion of your antigen in your antibody class will always be affected via your size of your particles. Kasi kung masyado kang mabigat, mabagal yung movement mo. Temperature can also affect that one class. And also the preparation of your gel, viscosity. Okay? Kaya dapat, kung 1% yung gel, 1% talaga yung gagamitin mo ka so that your antigen and your antibody can move easily. So what you need to do per method for your passive immunodiffusion is to always read the package insert class. If the package insert is that you need to prepare a 1% gel, prepare 1% gel because the 2% gel may not allow the movement of your antigen and or antibody causing a tama class. False negative result. Sino may kasalanan? Ikaw, medical technologies class and or medical laboratory scientist. Di ba imagine, mali mo lang yung pagka-compute ng preparation mo with a gel for your passive immunodiffusion, it can cause death to your patients. We need to be more careful, guys. I hope that you realize how our license is very crucial, okay, in dealing with patients' lives. And of course, amount of hydration. This is related to the gel viscosity and the interactions between the matrix and then the reactants. The matrix is with a gel that may have your antibody and or your reactants that may have your antigen. So with your passive immunodiffusion class, okay, have your single immunodiffusion. Okay? Kasi ano diba? As I mentioned earlier here, meron tayong passive immunodiffusion, electrophoresis, at saka light scattering. Under your passive class, immunodiffusion, meron tayong tatlo. You have your single immunodiffusion, radial immunodiffusion, at saka double immunodiffusion. So I'll be now presenting your single immunodiffusions. If you can observe, single, so one lang sa class na movement. So you allow the antigen and then the antibody to meet in between to form a precipitating bond. So for example, here in the tube, okay, so you have your agar gel here, and the agar gel is embedded with your antibody. So meron na siyang antibody class sa gel mo. And then, you have your sample, patient sample. Diba? Here, hindi, bigay na lang tayo ng example kasi I'm more on the infectious disease. I'll give an example that is related to infectious disease. So, example, you have your hepatitis C. Okay, so you want to determine if your patient has a hepatitis C antigen. Class ha, yung pasyente natin tinitignan natin if kung merong virus. Hindi antibody yung tinitignan natin dito class ha, antigen. Kasi through this one, yung tinitignan natin is that the antigen is the patient sample. So what we need to do is we prepare on the tube the antibody. Of course, this one is your hepatitis C. Kaya antibody embedded on your gel. Okay? Then from the patient, you collected the blood, for example, and then you centrifuge, and then the antigen, which is the virus, will be floating because they are lighter, diba? When you do the centrifugation, the virus is more lighter, then andun sila sa supernatant. You collect the supernatant, for example, either by plasma or serum, and then you added that one here. You allow the antigen, if kung meron, for example, this is your hepatitis C, will allow to move via diffusion. And then, your antibody also will be attracted to the antigen. Diba? So, if the antigen is present, the antibody will be attracted. They will move class and they will meet here on the middle. And then, kung nag-meet na sila class, then there will be a immune complex. Tama? So, if you have an immune complex, we have a precipitating band. Precipitating band, it means na this is the line where, or this is the band where the antigen in your antibody meets already. Okay? So, we have the single diffusion. We also have an agar base here. Nasa plate class na gel. Gel pala to class. Okay? So, here, if you can observe, you have your, for example, 2% gel here. Okay? So, you have your gel here. Ang gagawin mo is that you need to make a bore here. Okay? So, ano siya class, circle. And then, you add your antigen. Then, you, on the other side also, you add your antibody. And then, you allow the migration of your antigen in your antibody on the center to form a precipitate band. Okay? Kaya nagkakaroon tayo ng ano class? Ito, precipitate band. This one is also precipitate band. And of course, when you do this one, it's too difficult to see the interaction, right? Then therefore, we need to stain this one. And then after staining, if you can observe the antibody-antigen interaction, then this is a positive result already. And that is your passive immunodiffusion. Then let's move to your radial immunodiffusion class. Okay, for radial immunodiffusion, from the word itself, radial, then we measure the diameter class. Okay, we measure the diameter of your precipitating ring. So, for example, you have your antibody in the gel, okay, and then you have your antigen in the well. So then therefore, class, if you can observe here, this one, the yellow one, okay, this is the gel. Then the gel contains the antibody already here. Tama? Then, meron tayong butas dito, class. Okay? After na meron tayong butas, we can add your, on the well, okay, class, here, the well. Then we add your antigen here. The antigen here, the antigen here, and the antigen here. Then therefore, class, the larger the precipitate ring, and or the larger the diameter of the ring, this is proportional to the concentration of the antigen. So, anong ibig sabihin yan, sir? Pag malaki daw yung class, diameter ng natin here, it means mataas yung concentration ng antigen. Are you familiar with this one already? Tama, class. Because in your bacteriology, this is the same principle with your AST, diba? For AST, naalala niyo yung AST class, then we measure the zone of inhibition. Okay, same with that one also. Okay, and then this can also be a quantitative approach using this one. Okay, and then we can identify and or quantify, class, your immunoglobulin level. Okay, next, aside from that one, class, with your radial immunodiffusion, you have your Mancini and your Fahy. Okay, so I know that you're familiar with this one because in the laboratory, I think that you perform your RID already. Okay, so you have radial immunodiffusion with your C3. Okay, so if you can observe your class, this is your plate for your RID. And then, sir, bakit siya color coding? Okay, color-coded yan sila, class, based on the analyte that you want to identify. Like, for example, I don't know if Still, now the code is the same. Like for example, the C3, identification of your C3, the color is color green or light green. I don't know where the blue and then the red, maybe other proteins naman to class. Okay? But anyways, you can always check the package insert of the RAD kit before proceeding to the experiment and or performing the method. So we are sure if kung ano yung antigen na focus natin dito class. Okay? So the difference between your Mancini and your Fahy is that the method with your Mancini is your endpoint. And then in your laboratory, if you perform your RID with your complement. For example, you want to quantify the level of your C3 on the patient serum in our plasma. And then we use your Mancini. Kasi endpoint lang siya. Okay, ibig sabihin class, 1 point lang natin siya nire-read. Okay, like for example, after 24 hours, after 25 hours na incubation, mini-measure na natin kaagad. Ayan. So, mini-measure na natin siya kaagad yung zone of diameter, sorry class, the diameter. Okay? Here, the diameter of your ring. Okay? So, the concentration of your standard on the x-axis versus the diameter squared on your x-axis naman. Compare with your Fahy naman class, it's kinetic. And then, uh, lower yung incubation period niya class 12 to 28 hours. Okay? So this is the mostly tinatanong sa mga pre-board exams and board exams class. Sino sa dalawa ang endpoint and sino sa dalawa yung kinetics. Then you need to memorize that one. Lastly, under your passive immunodiffusion class, you have your double immunodiffusion. So, compared with the single immunodiffusion, we have only one antigen, one antibody. Correct? When we say double immunodiffusion class, in the case of this example, you have one antibody, class. You have your one antigen or one antibody, and then you have your uh, antigen. Okay, so the antigen antibody patterns form in Ochterlony double diffusion. One best example of your double immune diffusion class is your Ochterlony. And your Ochterlony method is very crucial in identifying your fungal antigen, specifically your Aspergillus. Kasi with your Aspergillus class, we have a lot of Aspergillus species, and you will try to discuss that one in your mycology and virology. So for Aspergillus, you have Aspergillus fumigatus, Aspergillus niger, Aspergillus, ano pa ba yung Aspergillus fumigatus, niger, yun lang yung maalala ko pas. Okay, so madami tayong Aspergillus actually. Fumigatus, niger. Okay, and then, like for example, here you have the class, three patterns of your Sterloni double diffusion. So you have your pattern of identity, you have your pattern of partial identity, then you have your pattern of non-identity. So let's start discussing your pattern of identity. So you have your A here, okay, this one, the A, and then you have your antigen here and antigen. For example, this one is your antibody class. And then your antibody will be directed to a different antigens. That's A and A. Okay, so for example, you have your Aspergillus fumigatus here, and then you have Aspergillus niger. Both are Aspergillus species. Then you have an antibody here. Your antibody here can also be can also identify your Aspergillus fumigatus and your Aspergillus niger. So when the antibody will try to migrate here, the antigen will also try to migrate here. Kaya nagkakaroon tayo ng precipitin line. And then the antibody also will try to migrate here, and then the antigen also will try to migrate here. Kaya nagkakaroon tayo ng pattern of identity. Always remember class, in the immunodiffusion, the main positive result is the formation of the precipitin band. Okay? So, ibig sabihin class, pag nag-meet sila nito, it means identical tong A here to A here. And also with here and then here. Okay? Kaya nagkakaroon tayo ng pattern of identity. Next, you have your pattern of partial identity. So, ano ibig sabihin ng pattern of partial identity class? So, ibig sabihin nito class that you have your A1 and A2. Then you have your A1 and A2 here. And then you only have your A1 here. Hindi siya A2 kasi A3 siya. So, ibig sabihin class, this one and this one will bind. Tama? We'll try to bind. Okay, so you have your A1, A1, A1. Ito. Okay? So, nagkakaroon tayo ng precipitin bond here. However, your A1, A3, your A1, A2 class, although they have a similarity with the A1, so nagkakaroon dito ng precipitin bond. Pero, the A3 here, kasi diba wala naman siyang A3 here class, kaya nagkakaroon tayo ng spur na part. Kaya kung meron tayong ganito na reaction, we call that one pattern of partial identity. So, ibig sabihin class 1 is identical to your antibody here, and then partially identical to the antigen 2. And now, let's move to the pattern of non-identity. Okay, with the pattern of a non-identity class, hindi siya nagkakaroon ng curve part. Kasi, yung A mo dito, at saka AB here, wala bind. Tama? Okay, nagkakaroon tayo ng non-identity here. Then dito din non-identity. Kaya, the result is pattern of non-identity. So, ibig sabihin class, the antibody here, like for example, this is your antigen A, antigen B, or sample A and sample B. And then, hindi siya related to your antibody. Okay? So, that is your double immunodiffusion. So, with single, mas madali. Diba? Kasi one antigen, one antibody. Tapos passive, you allow the migration of your antigen in your antibody and meet that one in the middle. Pag radial immunodiffusion naman class, we measure the diameter. Okay, so meron tayong Mancini at saka Fahey method naman class. Then you also have your double immunodiffusion kasi dinawa siyang double because you have two antigens in one specific antibody. And we will try to identify if they have the three patterns. Kaya pattern of identity, pattern of partial identity, and or hindi talaga sila identical. Then let's move to your electrophoresis class. Okay, so since we're done with your passive immunodiffusion, then next we will discuss your electrophoresis. So you know na already the difference between the two kasi diba kanina you have your passive immunodiffusion. Ibig sabihin, you allow naturally the movement of your antigen to your antibody to meet and produce a precipitin light. But here, this is induced by electricity. Kaya nagkakaroon tayo ng electrophoresis. Okay, phoresis, ibig sabihin separation. Then, the electro is the force that will try to separate kung ano ang meron sa sample natin, class. Okay? So, you have three types naman, class, of your electrophoresis. You have your immunoelectrophoresis. Next, you have your rocket immunoelectrophoresis. And also, you have your countercurrent, okay, immunoelectrophoresis. So, let's start with your immunoelectrophoresis. This is very simple, class, because if you can observe in the figure, you have your antigen mixture here. Okay, the antigen mixture is on the preparation orange. Ito. So, ibig sabihin, class, ito yung gel. Okay, this is the gel. Itong puti. Diba? Then you add your sample here, itong orange. Okay? So, wag niyo muna itong i-mind class, itong other colors. Kasi, yung first step daw is you add your sample here. Tapos, through the electricity, meron tayong movement class. The movement will differ if kung cation siya or anion. Okay, so nagkakaroon tayo ng separation. So for example, cationic itong nandito, anionic naman ito dito. So the proteins here that present on your antigen will move depending on the electric charge. Kaya nagkakaroon tayo ng different movement class. Okay? So red alert tayo dito class, typically the source of your antigens is in your serum. So from the patient serum, nag-aantay ng antigen. Then we allow that one to electrophoresis for rise, ayan, or to move or to separate. And then after that one class, pag nag-move na, we add your anti-serum. This is the blue one here, the antibody. Okay, and then added to the trough. Okay, on one or both sides. But usually class 1 side lang yung tinitingin. Nag-a-add tayo here. And allowed to diffuse. Tapos, pag diffuse nito dito class, they will, how can I say this one? They will bind to the diffusion of your antigen. So for example, this one is your antibody migrations. So for example, this one is your IgG, this one is your IgM, this one is your IgE, this one is your IgA. Tapos the antibodies are specific on that one. Okay? And then, pag nag-bind naman sila class, nagkakaroon tayo ng precipitin ring. Okay? So this is the precipitin ring. Kung saan yung edge ng diameter ng specific antigen natin class, nagkakaroon siya ng line. Okay? So these lines or arcs can be compared in a shape, intensity, and location to that of a normal serum control to detect the abnormalities. So for example, ito yung patient sample mo, dapat meron ka ding standard. And then we try to compare is, for example, ito sa patient sample, masyadong maliit compare with the control. Then therefore, maybe this protein is deficient to the patient. So the purpose of your immunoelectrophoresis class is to identify immunodeficiencies. If there is no precipitin, bond is formed for a particular immunoglobulins. For example, nag-add ka dito, tapos walang diffusion. So, walang precipitin ring. So, after nyan, kinumpirm mo sa control na meron, then therefore, okay, the patient has a specific deficiency on this type of antigen or protein. Okay, so deficiencies in complement, for example, can also be detected. Next is if you have your immunodeficiencies, you also have your overproduction of your serum. So kabaligtaran, we can also observe that one. So normally dapat ganito yung size niya class, and then with the patient sample. So for example, this is your control, dapat ganito yung size niya. But with the patient antigen class, mas malaki. Then therefore, we can identify an overproduction of a certain serum protein. In the identification of your monoclonal protein, free kappa in or lambda, okay, light chains may be used to identify your urine proteins. Okay, and then testing normal and abnormal proteins in serum or urine, and also we can use your immunoelectrophoresis to check the purity of your antigen. Okay, so for example, dapat meron ka lang one type of antigen. Okay, with a patient na dapat ma-identify. Pero meron siyang nagbigay siya ng different na forms of your antigen. Then therefore, maybe the antigen is not pure. So you can use that one. At saka dito naman class of identification of your monoclonal protein. You have your free kappa and your lambda light chains. You're familiar with this one, right? Because you discussed already with your clinical microscopy. And this one is, anong disease nga to class? Correct. Yeah, for multiple myeloma. Okay, so if you are still familiar with that, I hope that you can still correlate that one with your clinical microscopy. And next, you have your countercurrent electrophoresis class. With the countercurrent naman class, actually, this is the same with your passive immunodiffusion. Kasi, meron tayong antigen, meron tayong antibody, tapos meron tayong precipitin sa gitna. However, class, this is guided with the electric current. Okay, kaya nagkakaroon tayo ng ganito. And I think this is more faster. Tama? Because the electric current will allow a faster movement of your antigen in your antibody. So you're familiar with this one already. I presented this one already, right? With your passive diffusion. However, in your passive immunodiffusion, walang electricity. Okay? Dito, meron. Okay, so the method is still the same. Antigen antibody migrate toward each other by electrophoresis, and then we will observe a band. So in actual class, ganito yung itsura niya. You have two wells here, you have your antigen and your antibody, and of course, this is the control, right? So dapat expected na anti-serum, so meron tayong band. The sample, negative. The sample 2, positive kasi merong band which is comparable to the control. And then the sample 3, meron din siyang band that is comparable to the control. And these methods is your counter current electrophoresis. Then let's move to your rocket. Okay, immunoelectrophoresis from the name itself palang class, you have your rocket immunoelectrophoresis. So you are expecting that maybe the result may form like a rocket shape. Always take note also with this one class kasi it's a board exam question, and board exam recall questions in your pre-board exam. Recall questions also, I have your lower. Okay, that's another name of your rocket immunoelectrophoresis class. Okay, so in this one, this is actually an adapted form of your RID, but your RID or your radio immunodeficient deficient class does not have any electricity involved. Diba, we allowed to incubate that one in a longer period of time, around 25 hours, or it depends kung ano yung gagamitin mo, if kung endpoint ka mo or kinetics, okay, just like your RID. However, your rocket immunoelectrophoresis, we use your electro here, electricity. Okay, so your electrophoresis is used to facilitate, okay, your migration of your antigen in agar. So the antigen diffuses out of the well, then precipitation begins. Okay, so, the same principle pa rin siya with the RID class. However, meron lang siyang electricity. Then the end result, of course, you will produce a precipitin line. Then, the conical shape, kaya nagkakaroon tayo ng rocket immunoclass kasi meron pala siyang conical shape that resemble a rocket. Okay, then therefore, class, the height of the rocket is measured. And then, the height of the rocket is directly proportional to the amount of the antigen. So, the higher the rocket, the higher the concentration of the antigen. Okay, so if you can observe here, of course, we'll start with the standard. Okay, so this is your, sorry class. So this is your standard muna class. Okay, here, this one. When we do your standard here, standard setup, the antigen, a known antigen should have an increasing antigen concentration. Okay, so you can see a pattern like this one. The higher the peak of the rocket, the higher the concentration of antigen. This is the setup class. This is the setup of your standard. Okay, so wala, hindi pa to siya patient sample. Okay, yung antigen well natin dito is the standard. We just wanted to know if the well is working. Why class? Board exam question. Tinanong class in your lower technique daw, what is the pH required? Okay, that is very crucial in the movement of your antigen and your antibody in the lower technique. So you have your 8.6. Highlight na natin to class, pH of 8.6 is a requirement. So pag nagkaroon na tayo ng ganitong pattern, now we are ready for the patient sample class. So for example here, so you have your antigen sample here. For example, patient 1, patient 2, patient 3, patient 4. And then we allow the identification of your specific diseases. Pag myeloma, so pag multiple myeloma naman class, diba meron tayong ah, ah, meron tayong overproduction of your kappa, tama ba? Na antibodies. So please try to confirm that one. And then if the antibody has a higher peak, then therefore at the sorry class, if the precipitin arc is higher, then therefore the concentration of your antigen is also higher. Okay, so screening tool for differentiation of more than 30 serum proteins and then major classes of your immunoglobulins. So, pwede ding class, this one is The gel is embedded with your immunoglobulin IgM. And you want to identify the level of the IgM present on patient 1, patient 2, patient 3, patient 4. Diba? Then you can also use this one as standard. Then kita mo dito class, the patient 2, patient 3 has a high concentration of your IgM compared with the standard. Bakit kaya? Kaya baka meron siyang multiple myeloma class. Okay. And or malignant lymphomas. And or meron siyang decrease compared with the control. Okay? So that's how we do your rocket immunoelectrophoresis. Lastly, for your precipitation class, you have your light scattering. In your light scattering naman class, meron tayong dalawa. You have your turbidimetry at saka you have your nephelometry. And in turbidimetry, you discussed this one already in your clinical chemistry. Your nephelometry, you discussed this one also in your clinical chemistry class in your instrumentation. Okay? So, as a recall, here for the precipitation, specifically with the light scattering. Pag sinabi natin turbidimetry class, it measures of the turbidity or cloudliness of a solution. So for example, this is your solution. So pag masyadong madami yung analyte, then therefore, okay, the sample will become turbid. So the measure or the measurement will be the reduction of the light intensity. So meron tayong light source. Kita tayo, if you can observe the light source here, class, the light source here are three rays. Tapos blocked by the turbidity or cloudiness of the sample. Then, nagkakaroon na lang tayo ng konting light pass through to the detector. Then therefore, the lower the light that is detected with your detector, then the higher the concentration of your analyte. Okay, so that is your turbidimetry. With your nephelometry naman class, okay, the detector is set to around 10 degrees to about 90 degrees, okay, of your light scatter. So compare with this one, na meron tayong straight line lang. What we do is that we check the light scatter naman around 10 to 90 degrees. And this is actually more sensitive than your turbidimetry. And using your nephelometry class, as discussed in our clinical chemistry, you can perform endpoint or one-point measurement and or kinetics. So we are done with your precipitation class. Those are your principles and or methods and techniques under your precipitation. So I hope that before we'll move to your agglutination, you were able to grasp the basic principles of your precipitation reaction. Okay. So now allow me to move forward and discuss your agglutination reaction. So in your agglutination reactions, guys, we have two stage process. You have your stage one, sensitization, where there is an attachment of your antibody to antigen on the RBC membrane. So for example, you have your antibody here, and then you have your antigen here, this one. Of course, this is your antigen, right? Then the red one is your epitope. And then the epitope will bind to the Fab region of your antibody, and we call this one sensitization. This is, are you familiar with this one? Can you recall this one, class? Naalala nyo ba ito? Saan nyo ito unang nakita, class? Eto. Di ba, we discussed this one already? Tama. During your hypersensitivity reaction type 1, di ba? It requires a sensitization. Di ba? However, the sensitization will occur with the FC region. Tama. Exposing with the Fab. Naalala? Because the sensitization in your type 1 hypersensitivity is with your basophil. In your basophil have a Fc epsilon receptor, tama? Okay, that will bind the Fc portion here, exposing the Fab region. Kaya na, sensitized na siya. Yan. Bumalik na naman tayo sa hypersensitivity class. Because it's almost the same naman siya na interaction. Okay? So, the sensitization is only the binding of your antibody to your antigen. After this one, class, pag meron ng binding, okay, it can now proceed to your stage 2, which is your agglutination or lattice formation. Okay? So if you can observe here, class, kasi meron siyang malaking carrier which is your antigen. Compared ng precipitation na they're very small, then requires a lot of your antibodies here. But here naman, class, kasi malaki siya, okay, so you can observe a visible agglutination. Okay, so formation of bridges between the sensitized red cells. This one is your red cells, and then this one is your APA2. To form, sorry class, to form the lattice that constitutes the agglutination. Then therefore, the goal is still to form a visible, stable lattice formation. Class K or agglutination. So the same with your precipitation class, that your agglutination also have a different factors that may influence the reaction. And that includes your ionic strength, the pH, of course the temperature, also the medium that we will use, and also the nature of your antibody. So then, ano ibig sabihin ng nature of your antibody, sir? So it means class that if you have your IgM that a lot of, have a lot of that has a lot of Fab regions, then malaki yung binding capacity niya class. Then therefore, mas kita yung visibility of the latest formation. So allow me to discuss the types of agglutination. You have your direct, indirect passive agglutination, reverse passive agglutination, and agglutination inhibition, and also your coagglutination. The coagglutination is a method that is inspired with your Staphylococcus aureus, and later I will try to discuss that one class. Then let's start to cover first your direct agglutination. Your direct agglutination is too simple. From the word it's direct. So if you have antigen, you have antibody, then it will bind. Okay, sensitization, and then lattice formation. Tada! Visible agglutination class. One best example of that one is your blood typing. Like for example, you have your type A. If you have your A antigen in your RBC, then therefore you have an opposing antibodies, which is your anti-B. If you're type B antigen here, then your opposing antibody should be anti-A. Tama? Then if you have your type AB, then therefore, wala kang circulating na antibodies. Okay? Your type O, then therefore, you don't have null, di ba? Your type O is a null, then therefore, wala siyang presence of your antigen here. Then, the circulating antibodies of the patient is anti-A and anti-B. So with the direct agglutination class is that you have here your, for example, in this picture, you have your type B. And then the type B will interact with the opposing antibodies, which is your anti-B antibodies. So what will happen? It will form an agglutination. Then there will be a lysis of your red blood cell. Okay, so aside from your red blood cell class, there also have a lot of particle antigens, and that includes your bacterium, parasite, and your RBC. So, for example, you want to identify that the patient contains or that the patient is infected with salmonella, then you can use anti-salmonella, then try to add that one on the patient sample, then if kung merong agglutination, then positive siya. So example, you have for serotyping of your E. coli, salmonella using specific anti-serum, serodiagnosis of your toxoplasmosis, and determination of your blood groups. Ito yung kasi yung pinakamadaling example class for direct agglutination. Next, you have your indirect agglutination class, or we call this one passive. An agglutination reaction that employs particles that are coated with antigens not normally found in the cell surface. Okay, so we have example here with the passive naman class. So you have your virus particle, okay, with the HSV antigen. The HSV antigen is bound to your sheep red blood cell. So sheep siya, okay, here, it's not a human blood, okay, kasi nakalagay naman dito, di ba, an agglutination reaction that employs particles, okay, particles daw class, that are not, that are coated with antigens that normally, with antigens not normally found in the cell's surface. Okay? So in application class here, you have your indirect heme agglutination test. Kasi the particle carriers could be red blood cell, polystyrene latex, you have your bentonite, and or charcoal. Okay, with your charcoal naman, diba class? I think you performed that one already with your syphilis. Okay, you use your charcoal there. Kaya meron tayong ano, diba? Laboratory procedure in your laboratory session. So in my presentation, allow me to use the red blood cell, red blood cell here. Okay, so you have your positive reaction and then you have your negative reaction here. Okay, so of course the positive reaction should be with agglutination and then the negative reaction should be no agglutination. Okay, so now we wanted to identify if the patient has a herpes simplex virus specific antibody. Okay, so herpes simplex virus is a Baltimore's classification group number 1 class. And therefore, there are DNA viruses. And we will learn that one during your mycology virology course next semester. Okay, so if we wanted to identify your herpes simplex virus, a specific antibody to your patient, what we need to do is to prepare a reagent specifically with your sheep red blood cell as a carrier. And then you have your HSV or herpes simplex virus antigen. Then the antigen here will serve as your epitope, okay, that will bind to your part of your antibody and then forming an agglutination. Okay, so if, for example, you have a specific epitope here of your HSP, and you will add that one to the patient sample that does not contain a specific antibody of your HSP, then therefore there is no agglutination. So aside from your HSV, your class, you can use that one to your rheumatoid fever, class rheumatoid factor, you have anti-nuclear antibody in LE, lupus erythematosus, you have antibody to group A Streptococcus antigen, and antibody to your Trichinella spiralis. Okay, so in short class, in your passive agglutination, okay, we allow the carrier molecule to absorb the soluble antigen, okay, forming this one, and then interacting with your antibody here from your patient. Kasi ito class, diretso na kasi class. Dapat pala in-explain ko ito una, diba? Bakit siya naging ganito. So before pala naging ganito class, you have your carrier molecule, you allow that one to be absorbed with your soluble antigen, okay, forming this one. So the soluble antigen here is your HSV antigen. The carrier molecule in this slide class is your sheep red blood cell. Then you will allow that one to interact with your antibody. So if kung merong antibody yung pasyente, then therefore you have your agglutination. Okay, next you have your reverse passive agglutination class. Okay, with your reverse passive agglutination class, sir, anong kaibahan ng passive agglutination from your reverse passive agglutination? Madali lang class. Kasi sa passive agglutination class, what we did in the previous slide is that you have a carrier and then we allow the carrier to be, we allow the antigen to bound to your carrier. Tama? Okay, pag sinabi natin passive agglutination, we allow the antigen, okay, the known antigen, to bound to your carrier. Then we will identify your antibody. Okay? So, ibig sabihin ng passive agglutination class, yung known natin is your antigen, yung unknown natin is your antibody. Pag sinabi kong known class, ibig sabihin yun yung reagent natin. Pag sinabi kong unknown, yun yung sample natin class. Okay? Then therefore, yung passive agglutination natin, your sample is your unknown is your antibody, your known is your antigen that is bound to your carrier. Okay, in the previous slide, I used your red blood cell. With your reverse passive agglutination naman class, hindi target natin yung antibody. Yung target natin na meron sa pasyente class is the antigen. So then therefore, tinawag siyang reverse because we allow the antibody to bound here to the carrier. Okay, so this one is the carrier. Okay, and then, pag nagkakaroon, class, para siyang sandwich din. Then, forming agglutination. So, ganun lang kadali yung reverse passive agglutination to your passive agglutination. Next, you have your agglutination inhibition naman class. Okay. Ito, this is one of the class, confusing part with the student. Kasi when you try to imagine agglutination, you always think about the complex, visible complex as a positive result. But in the case of your agglutination inhibition class, the no inhibition pertains to a positive result. Okay? So the principle behind your agglutination inhibition class is that the reaction are based, okay, on the competition, okay, class. Ibig sabihin may competition tayo dito between the particulate and soluble antigens for limited antibody binding sites. Then the lack of agglutination is a indicator of a positive reaction. So highlight natin to class. The technique is called, okay, have here heme agglutination inhibition. If the particle in the reaction is RBC. Diba? So, ang sabi daw dito, class, so tinatawag lang natin na heme agglutination inhibition if the carrier is red blood cell. Kasi heme. Okay, so we use this one with the RBC. Okay, so if you can observe here, class, allow me to explain this one with the panel A. With the panel A class, if you can observe, you have your red blood cell component here. Then, walang interaction kasi red blood cell lang naman, di ba? May kasama pa siya dito, class. Wala. Siya lang mag-isa, di ba? So, kung siya lang mag-isa dito, class, then therefore, no reaction. Yun yung control natin. Okay, no agglutination. Okay? No reaction or no agglutination. But here, class, on the panel B, what we did here is that first, we interact the red blood cell to your virus. Okay? So when we did the interaction with your virus here, what will happen? There will be a heme agglutination class. Okay? Then your panel C naman class, you have here. So you have your antibody. Okay? Dito kasi class, known pa rin to class yung panel B. Hindi ko pala nabanggit class. Yung ano natin dito is the antibody. We wanted to identify if the patient is carrying an antibody specific to the antigen. One best example of this one, class, is your enteroviruses. Like for example, your polio. Okay, this is one of the best method to identify your polio. Okay, kasi yung ina-identify natin sa polio class is the antibody, hindi yung virus. Kasi we have a known virus of your polio as a reagent. Okay, so for example here, on the panel, yung panel A natin, red blood cell lang. So, dapat no agglutination. Yung panel B natin, meron tayong known virus. This is your poliovirus, a known reagent. And will bind to your red blood cell. Okay? So, walang antibodies, di ba? Walang antibodies dyan. Okay? So, walang antibodies from the patient. So, we allow the interaction of your red blood cell to your virus, okay, forming a heme agglutination. Then, therefore, pag merong agglutination, that's a negative result. Kasi wala ka naman talagang enough antibodies. Diba? So in your panel C, what happened is that, same with your panel B, meron tayong virus, meron red blood cell. However, we added the patient sample class, which is your antibody. So what will happen? The antibody will try to neutralize your virus. Okay, so what will happen, class? Pag na-neutralize na yan, the virus cannot infect or bind to the red blood cell. Kaya, wala siyang inhibition. Kung wala siyang inhibition class, then therefore the antibody were able to opsonize, neutralize your virus that is specific, okay, to your antibody. Okay, so then therefore, kung poliovirus to class, then nag-bind siya dito and neutralize, then ang pasyente positive with your polio. Okay, aside from your polio class, we can also use this one for detecting antibodies to your influenza. And last, you have your co-agglutination class. Okay, with the co-agglutination, the name gives to system using bacteria as the inert particles to which antibody is attached. And the bacteria that we use class is your Staphylococcus aureus. Remember, during you had your discussion in your Staphylococcus aureus, one of the virulence factor of your Staphylococcus aureus is your protein A, because your protein A will bind the Fc portion of your antibody, okay, exposing the Fab region. Okay? So if you can observe here, this is your Staphylococcus aureus, tapos meron tayong protein A class. Then the protein A has a specificity for the Fc portion, hindi sa Fab. Kaya ganito yung itsura niya, class. Dito nag-start yung idea with the co-agglutination. Sabi nila, oh, why not we will use your Staphylococcus aureus? Pero co-11 strain yung gamit nila dito, class, which is a virulent strain. Okay? And then, we allow to identify your antigen here, class. Then, pag na-identify na yung antigen, tapos meron na siyang agglutination, then positive. And mind you, class, that the Staphylococcus aureus will only serve as your carrier. So, ibig sabihin, class, we can modify this one, the protein E. Like, example, you can modify your protein A that will bind to a specific antibody, right? And then the antibody, for example, you want to identify your hepatitis C. Okay, so this one is a hepatitis, pero pangit pala, class, kasi bacteria yung itsura dito. Like for example, you want to identify your Yersinia intercolitica. Then this antibody will bind to the Yersinia intercolitica here. Then the Fc portion will bind to the protein A, exposing the Fab region that will bind to the antigen of your Yersinia intercolitica, okay, producing a particle agglutination. So that is your co-agglutination class. We also have your agglutination antibody by quantification using your titer. So what we do here, class, is the titer, okay, the maximum dilution that gives a visible agglutination. So uh, you're familiar with the dilution, right? Like for example, this one is the highest concentration of the dilution. Okay? So ibig sabihin, class, ito yung pinaka mababa yung titer niya. Okay? You need to dilute that one into twofold. So twofold dilution siya, diba? 2, 4, 8, 16, 32, 64, 1, 2, 8, then 2, 5, 6. Okay? So here, class, the endpoint is the well with the lowest concentration of the virus where there is a heme agglutination. Sa 521 class, wala na. Ito, meron pa. So then, therefore, the HA, the heme agglutinin titer of this virus is in the row 256 kasi 2, 4, 8, 16, 32, 64, 1, 2, 8, 2, 5, 6, or 2 to the power of 8 kasi 1, 2, 3, 4, 5, 6, 7, 8. Okay? Or 1 is to 256 dilution. Okay? Then, it depends on the heme agglutination natin, class, kung ano yung titer na positive siya. Okay? With your specific infectious agent. Okay? So, this is very simple kasi the method is only twofold dilution, and then you will only check the last well na merong agglutination. Then that will be your titer. Okay, that's it for your immunology series lecture 1. We're able to discuss your chapter 8 and then chapter 9, which is your precipitation in your agglutination. So I hope that you somewhat grasp the basic principles of the two very important serological principle, so that you can apply that one in the laboratory, and then you can also apply that one when you become intern, and then soon you'll become RMT. Again, if you have some questions in our clarification class with a discussion, please don't hesitate to contact your professors so we can clarify all the gray areas of your serology. And thank you for watching the video lecture, and God bless.